Hello and welcome to the 2.4 syllabus for this IB biology course, with this being proteins. Now, it's important to first talk about amino acids, considering they are what make up proteins in the first place. We know amino acids are joined by these covalent bonds. We know that two amino acids joined together are called a dipeptide. Now, they're bound together by what's called a peptide bond, which I'll talk about in just a moment. Here's our structure for an amino acid. We know we have that carboxyl group and that amine group, along with that R variant group here, which is what makes each individual amino acid unique. Looking specifically at this dipeptide and peptide bond, we can note that it's formed via a condensation reaction. I've drawn two amino acids here, as you can see, and we can note specifically this hydroxyl group and this hydrogen. As they bind together to form a dipeptide and a peptide bond, water is a product because we have too many oxygen and hydrogen molecules for when they bind, so therefore we have water as a product. When they bind, what occurs is as follows. We can see that the dipeptide bond, which I've highlighted in red is situated here. And we know that water has to be taken out in order for this binding to take place, so water is separate. And this is effectively your example of a dipeptide. An important skill to have is looking at the number of peptide bonds when looking at things like chains of amino acids, considering they typically come in long and complex structures. An easy way to determine this is just using a simple n-1, where n is your number of amino acids. So an example of this would be having something like 5,000 amino acids in a chain. So using this formula, we can we can tell that there are 4,999 peptide bonds. And just as a visual, we can see that in the instance where there are 5 amino acids, so 1, 2, 3, 4, 5, there are only 4 peptide bonds. 1, 2, 3, 4. So this formula is a simple yet completely valid way to determine how many peptide bonds are present in an amino acid chain. Some important key terms surrounding amino acids are as follows. We have our dipeptides, which I talked about earlier in terms of that dipeptide structure with that water forming here. Next up, we have oleopeptides, which are your 2 to 20 amino acids, so much larger than your typical dipeptide. Finally, and perhaps most commonly for proteins, we have polypeptides, which are effectively a polymer, so we can see that poly present in this word, meaning there's a chain greater than 20. So you're looking at a long and typically complex chain of amino acids when they're being referred to as a polypeptide. So amino acids have different types, with there being 20 overall present. Firstly, we have the essential amino acids, and we know that these cannot be made by the body, with their source for us being from food, so that nutrients we get from food provides these amino acids. Next up, we have the non-essential amino acids, which is where the body produces them, so we don't need them from that food source. Finally, we have the conditional amino acids, which are essential, so meaning that they do need to be from food in times of illness and stress, so not necessarily all the time, more so just during specific times is when that they will be required in that essential sense. So properties of amino acids, we know that they are colorless. They have a fairly high melting point of 200 degrees, with them being denatured at those high temperatures, so losing that structure that they have. We know that they're soluble in water, and we know that they are imperative for this idea of proteins. Now, it's just important to note also that the R group can ultimately change this these properties of amino acids, making them more unique dependent on that R group. So things like pH would be fairly subjective to this R group, as an example. So we know amino acids are imperative for proteins. Proteins are an essential part of life, with hormones, muscle, organs, things like our nervous system, all require amino acids in order for their successful function. It's important to note that amino acids are synthesized on ribosomes, more specifically translation, which I'll touch on in a moment. Now, we know there are a diverse, very, very diverse array of the orders of which amino acids can be placed. Obviously, there's 20 amino acids, and this formula effectively shows the possible amino acid sequence and how it is extremely different dependent on how many are in a chain. So as an example, for a 5,000 chained amino acid sequence, you're going to have a massive number of 20 to the power of 5,000 possible amino acid sequences. So ultimately, this shows how there can be a massively diverse array, huge diverse array of proteins. This number is massive, and as a consequence, it shows that these amino acids can form diverse arrays of proteins accordingly. So in terms of the process of protein formation, I'll touch on this briefly. We have firstly transcription. DNA is unwound with helicase, so we know that's the enzyme. An RNA polymerase comes in to make this complementary mRNA, which is then used later in translation. We know that the RNA, so the mRNA, we'll let that be an M, moves into the cytoplasm to those ribosomes. So typically your free ribosomes, which are in the cytoplasm. Next up, we have translation. Now, mRNA, we know, is read by your ribosomes, and each codon, so we know a codon is effectively 3 bases, is read, and a complementary tRNA is brought onto that ribosome, coding for 1 amino acid. So effectively, it's just continually read until we get to that point where we get that stop codon, and that's when you have your completed structure, which will ultimately end up in your 3D more complex form, and ultimately your final protein. So we know that proteins have a diverse array of functions, and I've just noted down here the perhaps most notable ones. So we have structure, hormones, immunity, so things like antigens, transport amongst cells, sensation, that idea of feeling, movement and locomotion, and of course the role of enzymes in the human body. Now, a useful way to remember the overall functions of proteins is this acronym, which once you spell it out is a good way to ultimately remember, and it's something which definitely has helped me. Moving on to some specific examples for proteins, we have Rubisco, with its full name being something I won't say now. But Rubisco is effectively seen in photosynthesis, catalyzing your first reaction of carbon dioxide in the chloroplast. We know it's globular. Now, a globular protein, globular and fibrous, I'll talk about following these examples, but they're effectively a more spherical type of protein. Insulin is another example. We know it's produced in the pancreas, and it's mainly involved in that blood sugar regulation. So we know people with diabetes who don't produce insulin have to find alternate ways to regulate their blood sugar. And again, insulin is globular in nature. Immunoglobulin is next up, and I've got a diagram here to help understand the shape. It's Y-shaped, but globular, so it's also spherical, and although this diagram doesn't illustrate it, it is spherical in essence, although it does have this Y shape to it. Now, immunoglobulin is involved in fighting infection for things like antigens. In this diagram, we can see that Y shape, that binding site for those antigens as well. The next example is rhodopsin, which is a protein linked to pigment cells. So we know they're involved with the eye, found on the retina. Again, a globular protein, and its specific function is to allow that very low light detection in the eye. So when there is not much light available, rhodopsin is what allows us to see. Moving on, we have collagen next up. Collagen is very commonly known as that structural protein seen commonly amongst the media and whatnot for skin, but it's also in your muscles and tendons as well, allowing that structure. Now, collagen is fibrous, and I'll talk about that later, but it means it's effectively elongated with the polypeptide chains parallel to each other compared to that more spherical globular. A final example is spider silk. Again, it's fibrous and quite elongated in nature, as you can imagine, produced by spiders. So I'm sure we all know a spider web, and that's effectively the protein that we're looking at there, that fibrous, elongated protein that is a spider web, or the silk from that spider web. So here is this comparison between those two structural aspects of protein. So fibrous typically has that structural role, so collagen, as that example earlier, and it's insoluble. So this, of course, meaning it can't dissolve in water, and also meaning that it is hydrophobic. As mentioned, it's parallel. Its polypeptide chains are parallel, so it's elongated. It's by no means globular, that's a simplification, but it's not that spherical type shape. So globular is soluble, and typically we can see things like a hydrophilic outside with a hydrophobic inside. Oops. So spherical, and we know it has a metabolic role. So just looking up earlier, rhodopsin was globular, immunoglobulin, globular as well, insulin, and Rubisco, all examples of globular structural type protein. There are different structures of proteins typically reflecting on their complexity. So firstly, we have your primary structure, which is effectively just the order and sequence of amino acids. As we talked about before, we know that there are peptide bonds between each amino acid. I've just drawn a quick diagram here to illustrate this primary structure. It's effectively just your amino acids, each joined by that peptide bond. Next up, we have a secondary structure, which is more complex than your primary structure, as the amino acids begin to interact with each other, folding and turning. So notably, we have our alpha helixes, and we can see that they adopt this spiral type shape, and this is due to the presence of hydrogen bonding on that backbone, which causes this structure. So hydrogen bonds here interact with each other, and therefore we experience that kind of spiral-ish shape, which is what we call an alpha helix. So they bond between the amino acids. And I'll just touch briefly on hydrogen bonding itself. We know hydrogen bonding in this particular instance would be bonding with oxygen. However, hydrogen bonding more broadly can be with nitrogen, fluorine, or oxygen. Similarly, another structure, secondary structure, is that of beta sheets, and they are different to alpha helixes in the sense that they have a different structure, so they have that more rigid type structure compared to that spiral type. However, we can link this back to our hydrogen bonding once again, and the hydrogen bonding is what allows this folding to take place. So ultimately, the folding can be different, so that spiral shape or that more rigid shape. However, that is the fundamental cause of the folding in the first place. Next up, we have our tertiary structures, which are a lot more complex. They are complex 3D structures involving that one polypeptide chain. Now, that's an important point to note before moving on to quaternary structures, but there is one type of polypeptide chain in there. And we know tertiary structures are effectively a combination of these foldings from your secondary structures. They can typically be globular in essence, and ultimately their structure is due to this R group interaction. So it's important to note that obviously each amino acid has its own unique R group, which is what allows that unique and specific protein structure for your tertiary structures. Next up, we have quaternary structures. Now, I mentioned before that tertiary only has that one polypeptide chain. Quaternary structures have 2 or more polypeptide chains. So in essence, they're quite complex. It's important to note as well that they may involve prosthetic groups. So I've given the example of hemoglobin and the heme group. So in this diagram, you can see that there are up to 4 different polypeptide chains there, but we can also note that iron heme group amongst the protein. So they are not polypeptide structures themselves, but they are a prosthetic group. So they may aid the process that the protein is trying to carry out accordingly. So that's your quaternary type structure. And it's your most complex form of protein structure. Next up, I'm going to talk about protein denaturation. When a protein is denatured, it loses its 3D shape. And if you think about a protein losing its shape, so let's just call that protein A, if it becomes unwound and it loses that interaction between hydrogen bonds, then in turn it will lose its actual function. So denaturing proteins is not good for their function because it cannot actually happen. We know protein denaturation is caused by 2 things. First, heat. Heat breaks those hydrogen bonds and, as I talked about before, manipulates that structure, therefore losing that function. Also, pH is something that can denature proteins. Now, we know pH affects the charges on the amino acids. So this in turn affects that interaction amongst the R groups, and as a consequence changes that protein shape, therefore having the same effect as heat and losing that structure and therefore losing that function. A final thing that I will talk about for the syllabus point is that of a proteome and genome. So a proteome is a collection of all the types of proteins that are in the body. We know it is unique to each person because it's determined by our genes, but it's also determined by environmental factors. So things like mutagens can manipulate the types of proteins that we have in our body. Looking at a genome, we know a genome is an organism's full set of genetic instructions. So the two are obviously quite interrelated in the sense that a genome is fundamental for a proteome as genes code for proteins. So it's important to know their relation, and it's also important to know that the proteome is substantially larger than your genome because there are an exponentially more amount of proteins that can be produced compared to the genes that we have due to that vast array of diversity and the array of different combinations that we can have with our genes, allowing for different proteins to form. So that's the end for this 2.4 syllabus point, proteins. Thank you for watching.